Welcome to the Pathways podcast from the American Society for Cell Biology. I'm your host, Mary Spiro. Today, we'll be learning about expansion microscopy, a technique of sample preparation that helps researchers to create higher resolution images of cells and tissues using regular light microscopy. Our guest is Paul Tillberg, who is a Janelia Fellow at the Howard Hughes Medical Institute's Janelia Research Campus in Ashburn, Virginia. Thanks for joining me today, Dr. Tillberg.
Thank you, Mary. It's great to be with you.
So let's start by talking a little bit about your background in electrical and materials engineering. How did that training help inform your development of this technique?
I would say the part of that training that was the most relevant was the materials part. And when I chose that combined electrical and materials engineering program, I think what I was really trying to do was get somewhere in the middle of physics and engineering, or really somewhere in the middle of different length scales. I like to think at different length scales, spanning from the molecular to the macroscopic. And in my time at UC Berkeley, in the materials part of that program, I worked in a lab, Ting Shu's lab, where they work with soft materials, meaning polymers. And the particular kind of polymers that we worked with there was block copolymers, which means that each polymer molecule has 2 or 3 different chunks, contiguous chunks, that have different properties, for example, hydrophobic and hydrophilic. And by doing that, you get phase separation, not at the macroscopic scale, but at the molecular or say 10 nanometer scale. And so what happens is that you can generate very elaborate nanoscale structures with ordering over micron or macro scale lengths. So that's probably the most relevant part of that experience for the later development of expansion.
But then you shifted into life sciences, biology, neuroscience. And at MIT, let's talk a little bit about how you and Ed Boyden and Fei Chen developed the expansion microscopy process there at MIT.
Yeah, we were thinking a lot about these new emerging super resolution techniques like STORM and STED and SIM. And these were all powerful and intriguing techniques to get more resolution out of the microscope. And so that was very interesting. And you could look at the physics of how they worked. That was fun to think about. But at the back of my mind, I always had this idea that maybe we could do it the other way. Instead of making the microscope more powerful, could we make the specimen bigger and then just use whatever existing microscope you have? And by thinking about that idea over a span of months and then bringing in that intuition about polymers and how they behave, I eventually had this idea that if you could just anchor the fluorescent things that you want to look at directly to a material that you engineer and synthesize everywhere in the specimen, and then dissolve the specimen so that it doesn't have mechanical structure, that then you could expand that engineered material and it would be like a 3D fluorescent expandable cast. So that would be a way of getting around the challenge, which is that tissue is very heterogeneous. So it's very hard to imagine taking something so heterogeneous and expanding it in a way that's uniform. So that was the insight, was that you could use this engineered material followed by digestion or dissolution of the tissue to drive uniform expansion. And then, and Ed was always game to brainstorm the craziest ideas about how to get from that little kernel idea of expansion to a final, an actual way to do it. And, I mean, he was just unfailingly encouraging of trying out ideas about the craziest way that, rationally would never work. But it's by starting to think about those things and try them out that you eventually get to the idea that does work.
What were some of the crazy ideas that he threw out there?
Well, another idea was, so one way that you can improve the Z resolution, meaning like along the axis to the microscope, which is the worst resolution. You get much better resolution in XY, you get worse resolution in Z.
So that's one way, depth of field?
Yeah, that's right. So that's always at least 3 times worse than your XY resolution for, even for the most powerful objective lenses. And for less powerful lenses, that ratio becomes even worse. But you can get around that, and people have done this by doing ultra thin slicing. So for electron microscopy, you slice in Z and you get, say, 70 nanometer thick slices. And so that's what you need to do transmission electron microscopy. But that also gets you a much better Z resolution than you would normally get with an optical microscope. So the optical resolution could be on the order of 500 nanometers, and that could be like the best you can do without getting to really crazy things. But by slicing at 70 nanometers, you can dramatically improve that resolution just by the slicing.
Right, but then you have to stack it back together.
Yes, you have to stack it back together. So getting a volumetric reconstruction is challenging. And it only really works in the Z dimension. But one of the ideas that we kind of tossed around was, what if we did that 3 times? What if we sliced in Z and then mounted those, and then turn it orthogonally, slice it again, mount those, turn it a second time, turn it, and then slice it in the other dimension. Maybe you could just physically slice it up into these 70 nanometer packets and then somehow handle them and separate them from each other.
Yeah, it sounds like a headache.
Yeah, fun to think about, maybe. Fun to do, definitely not. But, maybe there's this idea of cutting the thing up, and maybe 70 nanometers is not the optimal level to cut at. Because you have to do it mechanically, but doing it enzymatically is, ends up being the way to go.
How did you come upon the polymer that you ended up using to expand the samples?
So the way all polymers have some swelling behavior in a solvent, including the polyacrylamide gels that everybody, kind of is familiar with from electrophoresis. But that swelling is modest. You'll get maybe 10, 20, 30% of a change in the length of a thing. To get a really large swelling factor, you need a polyelectrolyte, which means that the monomer you use to make the gel is a salt. And what that means is that you have one charge, in our case, a negative charge attached to the polymer backbone. And then there's a positive charge that comes along with it, but it's free to diffuse around in the volume. So that is the only mechanism to get really big expansion factors, the kind of thing that you would use to drive, improved resolution. And the recipe that we use, essentially, we started with a electrophoresis gel recipe and just swapped out the non-electrolyte acrylamide monomer with the electrolyte acrylate monomer. And the reason for the acrylate monomer is that it is the single most well-characterized polyelectrolyte material, acrylate. It's the original polyelectrolyte that polymer physicists explored back in the 70s. It's used for diapers, of course, which I was not aware of that at the time, but it's the case. And also our favorite example of those swellable dinosaur things that, kids can play with. And they had a great parody of that in The Simpsons.
I think I missed that episode. So let's walk through the actual steps of preparing the sample. What happens first? Sorry. Let me back up just one moment. So I explained that beginning part of the idea and how it works conceptually. And then I want to make sure before we move on that I come to Fei Chen's contribution, which was the very first time that I tried to embed, biological cells in one of these swelling materials. Of course, it looked terrible. There was nothing that looked pretty about it. But I remember Fei came over to the microscope and he looked at it on the screen. It was digital microscope. And even though it looked like complete garbage, it snapped and he was like, oh yeah, this is the real deal. We could maybe actually get this to work. And from there on out, it was just the two of us, side by side, and we got it working. It was a super fun experience to do that together as a team. And, Fei was always extremely gifted and talented at designing the right experiment to get to the next step. What was it that he saw in the potential? What was it that led him to believe it would actually work, even though you say your sample looked terrible?
Yeah, well, it certainly wasn't the quality of the image, but I think there was the cumulative effect of, I had been excited about this idea and thinking about it and just kind of, tossing around ways of doing it for months. And I don't know, I think it was maybe the conjunction of having just that continued excitement from my end, coupled with, oh, there's something is happening here. We're doing something. It gives you a little opening. I think that's what made it real for him, to just see that you could do something with this in the real world at the bench on the microscope, coupled with months of enthusiasm.
Can you explain the exact process that you use to prepare a sample?
Yeah. So you start out with your biological specimen. It could be cultured cells, or it could be a tissue slice prepared by completely standard methods. You're going to fix with, again, completely standard methods of formaldehyde, maybe glutaraldehyde, but we usually worked with formaldehyde. Then you would do an antibody stain, again, completely standard. You permeabilize. Usually you put on first a primary antibody that binds to your target of interest, wash off the unbound stuff. Then you put on a secondary antibody that binds to the primary antibody and has a fluorescent molecule attached to it. That's what you actually see, is that fluorophore. So all of that part, that's all completely standard by the book methods. Now what comes next is the expansion part. And this is kind of the version 2 that people actually use now. I'm not going to get into the original way we did it. The way that people do it now is you put on a small molecule. Usually people use a molecule called acryloyl X or ACX. That molecule has, on one end, it has an NHS ester, which is a very commonly used chemistry that attaches to primary amines, for example, the lysines on proteins. So that end of this molecule reacts with basically all the proteins in your specimen, and whatever else has a primary amine, some fraction of those are going to get reacted and decorated with this ACX molecule. On the other side of that molecule is an acryloyl group, which is a monomer of the polymer that we're going to form next. So what we have now is essentially every protein in the specimen is now decorated with acryloyl groups. And those acryloyl groups can polymerize with the sodium acrylate and acrylamide that we put in next.
You know what this reminds me of? It kind of reminds me of when people try to preserve spider webs and they spray them with hairspray.
Oh, I love where this is going.
So it's, and obviously once it's sprayed with the hairspray, the web itself is, fixed in that state. And there's really, there's not a lot you can do with it, but it's just one of those ways of like kind of preserving that structure for at least a brief period of time.
That is very cool. And you could imagine making a hairspray analog that is swellable. I mean, maybe it wouldn't, like if the hairspray actually formed a swellable gel, then you could swell it, but you probably would not preserve features on the length scale that you would kind of need swelling for. But well, anyway, sometimes it's fun to think about crazy things. I also think about those ant colonies underground. I don't know if you've seen these videos where they'll like pour some molten material into the ant colony and it fills the thing up and then solidifies. And then they can dig it out and they get this full 3D representation of the interior of the ant colony. And it's incredibly complexly structured.
Mm-hmm. Too bad for the ants, but yeah.
Yeah, you got to feel bad for the ants. Got to feel bad for the colony there.
Yeah. Okay, so you've got this, the structure is decorated with the polymer, the acrylic. The monomer. Okay.
Yeah, the acryloyl monomer. The next step, and by the way, the specimen has been permeabilized already, because whenever you do an antibody stain, you have to permeabilize, which means you punch holes in the membrane. So normally a lot of these chemicals that we use would not necessarily get across the cell membrane, but we have essentially holes punched in the membrane. So all of the small molecules, these are all small molecules. I mean, we're talking like 100 Daltons, 10 to 20 atoms, small molecules.
Really
they get
small.
in, yeah, very small. They get in readily to permeabilize tissue very quickly. So the next step, we've got our proteins decorated with acryloyl monomers. The next step is we wash in the monomer precursor solution that is going to form the polymer. And the monomers are acrylamide, which we know from polyacrylamide electrophoresis gels, and sodium acrylate, which is the original swellable gel monomer. We also have a crosslinker that is going to form chemical links between chains. That's how you get, instead of just polymers, a gel with a 3D structure to it. And then we also have a polymerization initiator plus accelerator that's going to trigger polymerization. And we also include a polymerization inhibitor. So that inhibitor is going to gobble up the initiator until the inhibitor gets consumed. And that gives you a lead time where you can tune it. You get about half an hour so that there's time for everything to diffuse into your tissue, if it's a thick tissue, before polymerization commences really at all. So now you can imagine that you've got your proteins inside your tissue. They're now fully bathed and equilibrated with this monomer solution, which includes acrylamide and acrylate monomers and crosslinkers. Then you put it up to 37 degrees, and that just boosts the rate of initiation a bit. You don't really need to do that, but that's how we tuned all the recipe components. So then the initiator starts to do free radical initiation. You're basically just creating free radicals. The inhibitor soaks them up until the inhibitor is consumed. And at that point, the free radicals created by the the initiator, start to trigger polymerization of your monomers. That's when these small molecule monomers, 10, 20 atoms, start to link up into polymer chains. And of course, these chains can become large. They become, 10 nanometer scale macromolecules. They could be tens of thousands of atoms in these chains, potentially. And as those polymer chains are forming, they are also reacting with the acryloyl groups that are attached to all your proteins. So once that polymerization step goes to completion, what you have is a 3D mesh of polymer molecules cross-linked by the crosslinker that we include, and also chemically attached to essentially every protein in your specimen through the ACX. Now, the next step, of course, we wash out any unreacted monomers and the unreacted things, and then we have to do a disruption. So if you were to take that tissue that has now been embedded in the swellable gel and you wash it in water, the swellable gel is salt sensitive. So you wash out the salt, the material expands. But if you just have tissue as it is sitting there, it's not going to expand uniformly. It's going to get ripped apart because it has elasticity to it. So the way to avoid that ripping is you have to essentially dissolve the tissue. And there's two ways to do that. One is to put on an enzyme that cuts the proteins into small fragments, and then those fragments can pull apart readily. The other way is to treat it with a high detergent, high temperature step, and that basically disrupts the non-chemical bonds and to some extent the chemical bonds between proteins. So that allows you to take single proteins and essentially pull them apart from each other. You have to worry a little bit more about how uniform is the expansion in that case compared with the enzymatic digestion.
Yeah, this was the part that really kind of made me stumble when I was trying to wrap my head around this, because I just felt like, how can you be sure that the expansion is uniform? How can you be sure that you're maintaining the same spatial arrangement of everything in the tissue or the cell or whatever it is that you're looking at? How can you know for certain that because even the universe doesn't expand at the same rate, right? So
That's right.
how do you know?
Essentially, you cannot know until you check.
And how do you
have to check on a case by case basis. And so my favorite example of checking carefully on this is there's a few groups that are now using nuclear pore complexes to check the uniformity of expansion, at least for the nuclear pore complex. So the great thing about that nuclear pore complex is it is highly stereotyped. So for a given species or cell type, and probably also across species, the nuclear pore is basically a donut with it's very protein rich and a very stereotyped structure amongst pores. And we know what the structure is from electron microscopy. So what people will do is they will label the nuclear pores. You will measure the macroscopic expansion factor of your gel, of your cells, and of the nuclear pore. And then what you find is that the nuclear pores tend to expand a bit less than the macroscopic tissue. And by a bit, I mean something on the order of 10 to 20%. So There can definitely be non-uniformities. And I think that you have to be careful when you use it. I would not use expansion microscopy to get the, 10 nanometer level structure of something like a nuclear pore if you really cared about, do I know it to within 10%? I think it's a better use case to say we kind of know what the overall structure of this thing is, but now we want to figure out where specific proteins are located within it. We have a structure, we want to figure out where the proteins are. And that said, even if you can have a different stretch factor for different compartments in the cell, you don't get things just wholesale rearranging. So that's an important thing. The topology is preserved, even if something is a little bit less expanded than you would like it to be, like the nuclear pore.
Yeah, I guess it depends on how much that matters, like you said, to what it is that you're studying.
That's right.
Okay, so initially, when you first did this, you were able to get a four-fold expansion, which I guess is how much a diaper will expand. And as anyone who's had children knows, those diapers can get extremely large.
By the way, the diapers, there's a lot of filler material in there. So fourfold gives you a 64x volume increase, which is not, thankfully, how much a diaper swells.
I don't know. I beg to differ. Some of those diapers pretty scarily large.
Yeah.
But so now it looks like you're at the 10x or the TRex.
Yeah.
Of
the dinosaur theme comes back.
Yes.
Right?
Yeah. So how have you achieved this? I looked at the paper. It looked like you played around with the recipe and temperatures and different concentrations.
Yeah. That's right. So the really important thing that controls the expansion factor is the crosslinker. So if you don't have any crosslinker at all, you don't get a gel. You get polymer molecules that are floating around in solution, and they will dilute to whatever extent they have solution to dilute into. Now you start adding crosslinker to it, and those polymers link up into a network. And the more crosslinker you add, the more kind of pulling force that network has. So you increase the crosslinker, you get less expansion. You decrease the crosslinker, you get more expansion. The trade-off there is that the less crosslinker you have, the weaker the gel gets. So in fact, in the original EXM paper, we explored this a little bit by reducing the crosslinker and going up to a gel that expanded something like 8 or 9x. But the problem is that that gel was too weak to even stand up under its own force of gravity.
That's so like having very, very loose jello. Like your jello just didn't set up properly.
Very loose. That's right. So if you take the water away that is floating in, it doesn't even hold its shape. It just kind of, it's got this blobby jellyfish thing. So we figure we're not even going to go there. That's not going to be good enough to preserve structure at any length scale if you can see that it's macroscopically deforming. So for the original paper, we basically just chose an intermediate crosslinker concentration that gave us 4x expansion, which felt like enough to be worth trying. And then we would figure out how uniform it really ended up being. So the trick is, can you decrease the crosslinker, but still have a material that's tough and easy to handle? And with TRex, we essentially just played around with mostly increased monomer concentrations. The way to offset that softness of the material from low crosslinker is essentially to increase the monomer concentration. The more monomer you have in there, the tougher of a material you're going to get. You can also make the material even tougher by re-embedding in another gel, a second gel. And that what's called an interpenetrating network ends up being much tougher than either network alone. That's another way to do it. But when you do that, you need to do a second embedding step, which is a bit of a hassle. So with the TRex project, we just wanted to see what's the limit that we can do with a single gel step, something that you could do on the order of a day or two.
And so do you think 10-fold is about the limit of where you can go?
I think so, personally. Now, that said, we only really explored a few parameters there of monomer concentrations, crosslinker, initiator. There's, of course, many, many materials out there that could be explored to try and push that further. But my feeling is that it's not going to go too much further than that. And at some point, you come up against a very hard limit, which is that the distance between crosslinks in your material is filled by polymer chains that are just flailing around randomly. You know, the conformation of a polymer chain is randomly sampling all possible conformations, i.e. not retaining spatial information. So at some point, if you make too loose of a network to begin with, you're just going to, if the crosslink to crosslink distance becomes 10 nanometers, then you're not going to have much hope of preserving any information below the 10 nanometer scale. Now, nobody has looked at that carefully. Like, we don't really know what that distance is, but at some point you will come up against a hard limit like that.
What types of tissues or cells is expansion microscopy most well suited for?
So brain is the easiest that we've seen. Brain tissue just tends to be very soft. You can also do cultured cells, of course, but it's with like thicker tissue slices that you really get a differential benefit compared to other super resolution techniques. So I'll focus more on those thicker tissue slices. So brain is very soft, so it's very easy to disrupt it fully and get uniform expansion. Other tissue types have things like collagen in the extracellular matrix. Collagen tends to get extremely tough when you fix it with formaldehyde. So it's hard to disrupt it fully, but you can. You basically just need to increase the harshness of the digestion step. And that could mean increasing the temperature. It could mean adding more detergent, as harsher detergent, digesting at a lower salt level so that you kind of get more swelling in the gel during digestion. So we think that gives better access to the enzyme. So you can adapt it to other tissue types. The other thing that can happen is that if you have, for example, some insect tissue that has chitin or something, people actually expand that by putting chitinase on the tissue. So if you've got a protein that our proteinase K enzyme isn't really digesting effectively enough, you can put on an enzyme that digests whatever it is that's holding your tissue type together. And people have shown that it actually improves performance.
Obviously, you can't do this in live tissue.
Correct. That would be interesting.
It would be a mess. And then you also described, there was one paper you sent me that described the Easyfish method that seemed to be for thicker tissue samples. Tell me a little bit about that.
Yeah, so the Easyfish, we started with the X-Fish method, which was spearheaded by Fei Chen in Ed's lab. And what that method let you do is basically retain not just protein in the gel, but also retain nucleic acids, particularly RNA molecules. And then you could come in with fish probes. Fish, for anybody who doesn't know, it's a way of probing for RNA. So you basically have, it stands for fluorescent in situ hybridization. You have DNA oligo probes that are fluorescently labeled. You put them on the tissue, they bind to their complementary RNA strands. And that's how you can localize individual RNA molecules. So what they showed with the X-Fish method is that you can also retain RNA in the gel. You can probe for it with fluorescent probes. You can amplify that using amplification methods like hybridization chain reaction, for anybody who knows about it, and that it works well in thick tissue. And what we just wanted to turn this into a workhorse that particularly people at Genelia, but of course everywhere, could just use routinely to do multi-round fish in thick tissue specimens. And the multi-round part is important because RNA tends to be a little finicky. It can degrade readily from RNases, which are very resistant to degradation. But it turns out that when you have everything embedded in this gel, it seems like the RNA is actually quite stable. And we're able to strip and reprobe over 10 times over the course of months and still have on the 95% of the RNA molecules are retained even after those lengths of processing. And we also did a bit of fine tuning and optimization to the method, a little bit of reagent substitution to make things cheaper and a little more convenient, and then showed that you can use this method to do 10 or more rounds. You get 3 different species of RNA per round. So we got on total on order of 30, several dozen transcripts, and that you can get extremely high quality images of these transcripts in that thick tissue context. What that allows you to do is if you have single cell sequencing data from your tissue type of interest, you can pick genes from that single cell sequencing data set to identify cell types that you discover from single cell sequencing. And then you can figure out the spatial arrangement of those cell types using your 30 chosen transcripts in that thick tissue context with extremely high quality imaging.
So once you've prepared a sample using this method, either Easyfish or any of the previous expansion microscopy methods, how long is it usable? Can you like you said, can you reprobe it for months and months, or how long does that go?
Months and months, definitely, probably longer. So we had about 5% loss after 12 rounds. We were doing 1 round per week. So that's 3 months, 5% loss. There's no reason to believe that loss rate would increase over time. It seems like it's pretty steady. So if you don't let the specimen dry out and you don't have a pandemic intervene, you could probably save these things for years.
You got to keep it in a little jar of water. How do you keep it?
Yeah, just stick it in PBS, salt solution, or to increase the stability, you can stick it in 70% ethanol to really keep things stable. The RNases don't like to operate in 70% ethanol.
Mm-hmm. Well, I really love the idea that these techniques focus more on the sample preparation, especially like, let's say if you've got a rare or unusual sample and you really want to, um, you don't want to chop it up a bunch of different times because you'll waste it. And it doesn't rely on the use of expensive microscopes. No offense to all of our microscope sellers out there.
Or chemicals, by the way. The chemicals are also very cheap.
Okay. Mm-hmm. Chemicals and the sample preparation. Do you see this as a way of democratizing imaging science?
Yes, definitely. Absolutely. That is, I think, one of the big hopes for this technology. I think the biggest caveat to that, however, is that when you expand as much as we do, you expand fourfold, that's 64x in volume, your signals get very dim. So for a sort of 3D extended structure, the signal will be literally 64 times dimmer and more like 100 times dimmer because we lose approximately half of the fluorophores. Now, it's not quite as bad as it sounds because the background also comes down dramatically. So your signal to noise ratio ends up being boosted. But when the signal is so low, that means that the signal level is a limiting factor. So you need to have a high quality antibody, and you might need amplification. And sometimes those amplification techniques might not be so cheap. So I think that the main thing that we need for the democratization to reach its full potential is to figure out how do we really get reliably good staining with very high signal, and not have to choose our favorite structures of interest that people love to demonstrate technologies like microtubules. If you want to look at your protein of interest, are you going to be able to stain for it intensely enough that you're still going to find it after this 100-fold signal loss?
Now, for our life science educators out there, could you imagine a method, a protocol that you could develop that might be able to be used in a high school classroom or a college classroom?
I think so, yeah. The acrylamide is pretty toxic. However, acrylamide appears in coffee and potato chips, so it's not as bad as some chemicals, like, alkylated mercuries, I guess.
Probably half the cosmetics I wear.
There's acrylamide in a lot of stuff.
Yeah.
So it's not like, you can't touch it, but it is something that in the quantities that we're using it at, you need to be careful. So I don't know what the restrictions would be around having high schoolers work with it. I think that you can have high schoolers work with this stuff safely, but that kind of comes down to what the regulations are on that. But the methods are definitely easy enough for somebody without extensive training to pick up. So you could definitely imagine doing this in a classroom setting.
Mm-hmm. Very interesting. So
the antibodies tend to be a little expensive, by
Yeah.
the way. So you might want to be judicious about choosing what you're going to stain for. Maybe you just want to use small molecule stains instead of antibodies. And then I could see a very cool, classroom demo being done.
Yeah, it's got the wheels turning. So what are some interesting discoveries that you've seen either from your own work or from other people's work using expansion microscopy?
So one example that comes to mind is from Josh Vaughn's group. He was a very early adopter of EXM, and they combined EXM with SIM, which is structured illumination microscopy. You get another 2-fold boost in resolution. So they use that to look at the cytoskeleton of Giardia parasite. And so these parasites have a very interesting cytoskeleton that is involved in whatever structural transformations they use to stick onto cells, and then whatever they do, it's involving the cytoskeleton. And there's a lot of information known about the elaborate 3D nanoscale structure of that cytoskeleton from electron microscopy. There is also information known about the proteins involved from more like genetic approaches. But what they were able to do was take those two and put them together to say, we know there's this cool ultrastructure from EM. We know that these proteins are involved somehow from other methods. And with expansion plus sim, they were able to see how are these proteins actually distributed in this known ultrastructure. So that's an example of, I think, a very cool use for it. Another example that I would like to mention comes from Josh Lilvis in Barry Dixon's lab here at Genelia, where they use, so we have, at Genelia, we've collected these amazing electron microscopy connectomes, where you can see every single synapse of every single neuron in an entire fly brain. Now that's incredible. It's an incredibly high quality data set, and people are going to be mining that thing for insights into cell biology and neural connectivity for years, maybe decades. But it's also just one fly brain. What we thought was, could we use expansion instead of electron microscopy? And we're not going to try to get every single neuron, but we're going to try to get defined subsets of neurons. And if we can resolve the connections between those subsets of neurons, we could get maybe a limited or a sparse connectome, but do it for a much greater number of animals. And so Josh has spearheaded using that technique, and this is out on bioRxiv now. They showed that you can count the synapses between genetically identified cell types. They agree with the results from electron microscopy, but instead of doing a single animal, you can do 10 animals, 100 animals. That allows you to now look at the comparison between male and female, variability between individuals, variability across experimental differences. And he also showed that these synapse counts correlate very strongly with physiological measures, like how strongly are two neurons coupled together in terms of their activity levels. You drive one, does it drive the other? Very strongly predicted by these synapse counts that we measure with expansion, and you can do it in dozens of animals. The other one is that in the Easyfish paper, we show a finer degree of parcellation of the hypothalamus than was known previously. So that comes about because we got that large 3D structure that really allows you to search for finer. And these parcellations, it's not like one region is super high in one gene and the surrounding material has none of it. You know, it's not that clean of a parcellation that it's obvious when you just first look at it with a single gene. But by doing the multi-round fish and thick tissue, we were able to tease out that finer scale parcellation of the hypothalamus. And so now, of course, people can follow that up and see what is the functional or the physiological significance of that.
And you also sent me the paper that showed the infected, human airway cells infected with SARS.
Yes.
Yeah, that was interesting. You could actually see it sitting in there.
That's right. That's right. So that's from the Capitaine group at Utrecht. So we essentially, this was the whole TRex project was a collaboration with that lab. They're like the beta user, and I developed the technique. And they went on to use the TRex method in primary human airway cells infected with COVID. And as you say, they show these really incredible remodeling of the cilia on the surface of the cell. And then they also have what to me is a really iconic image in that paper, where they show that they label the membranes of the cells, and you can see that the virus is packed into multivesicular bodies, which are larger than normal. And then they caught one of those multivesicular bodies in the act of fusing with the plasma membrane. And you can see a little hole where there's a continuous space between the extracellular side and the lumen of that multivesicular body. So, I mean, it's just a stunning image. I love that image. You can see little virions in there. You can see the multivesicular body membrane. You can see it fusing with the plasma membrane.
And what's the scale? What's the length scale of something like that?
Yeah, I think, well, the virions, I think, are, what are they, like 50-ish nanometers? And that multivesicular body must be on the hundreds of nanometers to micron scale. I'm kind of guessing from memory here, but I think it's something like that.
Yeah.
Normally what we do is we say, okay, we know the expansion factor for this recipe is about, 10x. Sometimes maybe it comes out 9.5, sometimes 9.8. But if you don't really care about the nanoscale distance to that 10% level, we just say, okay, look, it's about 10. And so we're going to divide all the scale bars by 10, and we're going to put a tilde in front of it, and that means approximately. If you really care about the super nanoscale precision, you would not do that.
Yeah.
You would be more careful. But for something like, for most actual use cases, that is sufficient.
Okay, so what do you see for the future for expansion microscopy? What are you hoping to accomplish? What's coming down the road?
Well, one thing, of course, is to further that democratization. I think there's a lot more, potential there than has been realized. And one thing is that it appears to be a fancy technique with, things that people may not, especially microscopists, may not be familiar with these kinds of specimen processing steps. But they're not actually that bad. You can pick it up pretty easily. So I think that people might be a little hesitant to try it out because they're not sure it's going to work. Maybe it's not going to work. Maybe I'm not going to know if it's working. So there's more work to be done there to, get the word out, get people to try it. The other thing is that there's a two-edged sword of being compatible with existing staining methods. One is that you don't have to change your staining method at all. You just do it and then expand it. But the other side of that is that whatever limitations you have, you're going to inherit those. The antibody staining is not going to be any better after you expand it than it was before. You're just going to highlight the problems, such as low density of staining. The efficiency of binding may be low. You may not even know what the efficiency is. The efficiency of binding an antibody could be different from place to place within a cell, could depend on the local environment of that protein. So all of those are problems that EXM does not solve, but by expanding and then staining after expansion, maybe we can solve those problems. And quite a few groups are now working on that and making, I think, great progress in the past couple of years. And that's where I think some of the most exciting stuff is going to come in the next few years, is to see, can we really demonstrate that we improve antibody performance by doing it after expansion?
Well, you can't solve a problem until you can see it, right?
Absolutely. That's right. Necessity is the mother of invention.
Is there anything else that people should know?
I guess the main thing that I would like to tell people is, firstly, try expansion. Just try it. And it's actually quite easy to get started if you just, buy the chemicals, make some gels. Don't even try to put tissues in them. Just make some gels. The whole process is quite robust. If you wanted to start with TRex, maybe increase the Crosslinker a bit so that you're going to get a less expanding gel, but it's going to be more robust. And you can change how you make the gelation chamber to make it fit your specimen type. We just cobble together materials in lab to kind of make it work. You can play around with it. So that's what I want people to do is try it out, play around with it. Once you get comfortable handling the material, then you think about applying this to your scientific questions.
Well, it sounds like a really exciting area to get into, to try. I really appreciate the time that you took to explain everything in such great detail. Hopefully people will be encouraged to give it a shot. Thanks so much for talking with me today, Dr. Tillberg.
Well, thank you, Mary. It's been my pleasure.
The Pathways podcast is a production of the American Society for Cell Biology. Thanks for listening.